In this module, we will talk about some misunderstandings that happen when we talk about the medium just for the industrial fermentations. So when we talk about in a microbiological point of view, there are different kind of the medium. Sometimes the student asks that can we use those medium for the fermentation that we mostly use in our microbiological books and microbiological studies. So here we want to know about what are those medium and what is the basic difference about the industrial fermentation medium and those medium. As we have already discussed about the defined and undefined medium just on the base of their component. Either the component consists upon a pure compounds. On the other hand, if the compounds are not, their definite composition is not known, and they are the from agriculture byproducts or industrial byproducts, exact composition is not known, then we give the name of undefined medium. So here you can see that when we call and when we see about the selective media, and then in other you can see that differential medium and then enrichment culture. So these selective medium, these three different kind of the medium that is mostly used while isolating different specific microorganism in microbiological point of view. So as you can see in this slide that selective medium are designed to suppress the growth of unwanted bacteria. So just by selecting a specific medium that will only facilitate to grow some organisms or a microorganism that can only facilitate a specific, and then they have a check over undesired microorganisms. So for example, in this slide you can see that bismuth sulfite is one medium used to isolate the typhoid bacterium, the gram-negative Salmonella typhi. So bismuth sulfite inhibit gram-positive bacteria and most gram-negative intestinal bacteria other than Salmonella typhi as well as. So the another medium that is suboride dextrose medium, so which has the pH of 5.6, is used to isolate the fungi that outgrow most bacteria at this pH. Sometimes we utilize starch that only help to isolate those microorganisms which has the capability to produce amylase or starch degrading enzymes. So selective media are mostly used in some cases in industrial fermentation too, because when we have to grow our microorganism in such a way that only facilitate our desired product in a maximum concentration. So in the absence of that ingredient or a medium, then we cannot talk about our product of choice. So the selective medium is only designed for our selective objective. In case of microbiology, that is utilized for the isolation of specific organisms, but in fermentation the selective medium is only used just to produce a very selective product. As concerned the differential medium makes it easier to distinguish the colonies while isolating the organisms, because when we culture the organisms on a differential medium, then there will be isolated colonies. We have a very good example of blood medium. In blood medium that contain the blood. So that microbiologists often use to identify the bacterial species that destroy the red blood cells, that human pathogens and those pathogens which can utilize that protein and that nutrients which present on that cells. So these species are Streptococcus pyogenes, So that bacterium that cause the strep throat. So differential medium is basically that give a specific character to isolate some specific organisms. But differential mediums having less use in industrial fermentation. But there is one or few examples we can quote, but mostly the differential media are mostly used in the isolating of some specific microorganisms. So as concerned the enrichment culture is mostly used at the time of the isolation of the microorganism. Sometimes when we want to isolate any microorganisms, when we collect the samples, in that sample, then there is not a sufficient amount of that organism. So we use enrichment culture, we add some extra nutrients, extra compounds, and we provide some extra in the same environment just to increase the population of that microorganism. So that's why enrichment culture is basically that kind of the medium in which we give an extra nutrient in enough quantity. That's why that is known as enrichment culture. So the cultivation of sometimes in case of aerobic fermentation, we have to supply the oxygen. But in case of anaerobic bacterium, that poses a very complex situation, because there are some microorganisms for which the oxygen is toxic. We have to remove the dissolved oxygen even too from the medium, just by using some compounds that reduce or having a chelating or sequestering effect on the oxygen that is present in the medium. So we have to use such compounds which eliminate the oxygen from the medium. You can see in this slide that one important example is the use of sodium thioglycolate that is mostly used for the elimination or for the removal of the oxygen from the medium, because it chemically combine with the dissolved oxygen and deplete the oxygen in the culture medium. So there are other reducing agent, sodium sulfide, etc. So these are some specific agents that give a specific environment that is required to the organism. So when we talk about the growth medium, so we have to be very clear about the isolation media that is used just for the isolation of specific microorganism for specific object or a specific criteria. On the other hand, there is industrial fermentation, and that is also having the difference from the lab scale to pilot scale and then subsequently to the industrial scale.